Hi, Fiona.
Hi, good morning, everyone.
Yeah, we're still waiting on a few people to join, so we should be starting any minute.
Yeah, I think we can give people another minute, and then after we have Simon joining in, and then we can start the webinar.
Yeah, and I just confirmed we're good to go on YouTube.
Yeah, and we're just waiting for Jesus from Enzyme, but he's last.
We can give people another minute, and then after we have Simon joining.
Oh, there's Anthony. Hi, Anthony.
Oh, Anthony made it.
Hey guys, sorry I'm late.
No problem. Yeah, I say we get to 11:05 and then we can get started because our two first speakers are here. So
Anya, I'm gonna promote you to host, just in case things goes wrong, because I've yeah. But we're good to go on YouTube. Let's see here, make host.
I think Jesus is online as a participant, so I'm going to try to promote him to the panelist. But I think we should be ready to go.
yeah, great. Okay, so I guess I can kick things off. Thanks everyone for joining another installment of our virtual biocatalysis protein engineering meetup series. This is a very special edition of our meetup series. Normally we highlight two academic speakers and one industrial speaker and have a mix of both, but in this session we wanted to do an entirely industrial speaker panel. So we have three different companies that are going to give you talks surrounding their work and their research. Normally when we do these meetup series, we kind of have unintentional themes that kind of emerge from the speakers, just because the nature of protein engineering and biocatalysis go so hand in hand. But this time we actually intentionally set out to kind of give individual stories from individual aspects of this field. So we have Enzyme, which is a computational, they do computational approaches for protein design and protein selection. We have Ginkgo Bioworks that is going to talk about their work in protein engineering. They do a lot of work in enzymatic cascade development, microorganism engineering. And then we have Prisomics, which develops research tools and toolkits around identifying new and interesting enzymes that do new and interesting chemistries. So I'm really excited to see kind of how all of these talks, while individual, fit together in kind of this broader scope of protein engineering and biocatalysis. And then after we have our talks today, we're going to take 20 minutes to do a brief panel discussion with all of the speakers, where they'll share their thoughts on kind of the current state of the field, what they're excited for, their own personal paths in this field. So if you stick around after the talks today, we should have a nice panel discussion with all of our speakers today. I would like to kind of go over the format of the session today. So we'll have 3 15-minute talks with 5 minutes for questions. If you do have questions, please use the Q&A function at the bottom of the screen on your Zoom window. If you put it in the chat, we'll have to go look in the chat, and it gets a little confusing. So if we can just consolidate everything into the Q&A window, that would be great. And Anya is going to field questions. If there's time, or it looks like the question is good, then we will promote you to speaker privileges and you can ask your question directly. So we'll get through to that. Finally, before we get started on the talks, I just want to do one minor plug for Merck. We're currently actively hiring chemists, biocatalysis, protein engineers, molecular biologists, people who have experience in microfluidic technology. If you're interested in a position at Merck and want to learn more, please reach out to Anya and I. We can help direct you to where you would need to go to apply, or if you just have questions, we'd be happy to help you out. So yeah, I think that's everything. I guess we can start with our first speaker today. That'll be James Finnegan from Prosomics, who's going to be introduced by Simon. So you guys can go ahead and unmute yourself, share your screens, and go ahead and get started.
Hi, good morning, good afternoon, and probably good evening. I'm Simon Charnock, Managing Director of Prosomics Limited. I'd just like to thank Anya for the invitation to speak today, and actually for suggestions that Anya's made about Prosomics as an emerging company in this field over the years. She's made some really nice and generous suggestions. And I have to apologise as well, this is my first meetup, but I think this format's a great format, and I'll certainly be much more loyal from now on. For those of you that haven't met Prosomics before, we're an enzyme discovery company. And at this stage now, we've invested more than 100 woman and man years, researcher years, in developing what's the largest range of natural maximum diversity biocatalysts. We have more than 5,000 enzymes now in this native panel. And we provide this panel to big pharma as a focus, but to lots of other sectors as well on free of charge basis. So we really only get paid when we get hits that go through to a significant use. What we're also into these days is enzyme production, because it may seem logical that as an enzyme discovery company, you would also produce enzyme in-house at scale and act as a CMO, but that's not something common in the field, which is quite surprising, but that's an area we're really focusing on now, and that's enabling us to take additional market share. Anyway, James will explain about the enzyme discovery side of Prosomics, and James is our general manager. He's worked his way up through the structure here at Prosomics. He's been here a long time. I've actually known him for a long time now, and he actually, before going to university, he visited Prosomics to do some work experience, and he was very good at bioinformatics. Even before he'd gone to university, he had a keen interest in it. And I said to him, if ever he wanted to do a little bit of work whilst at the university, he'd be very welcome. And eventually, 6 months later, James contacted and said, oh, can I do some work? And I said, oh, we haven't really got any bioinformatics to do at the moment. And he explained that he was so poor at the time, he didn't even have any money to eat. So from time to time, we engaged him at university, eventually came back to Prosomics. And now, as I said, he's our general manager. So I'll pass over to James. James?
Thank you. Yeah, I'm James. So I'll quickly start off. As Simon said, we're a small enzyme company. We're based in North England, and back in 2008, 2012, we were an enzyme discovery company fairly strictly. 2012, 2016, we started to manufacture 1 gram to 100 gram scale in-house. And then since 2017, we've been pushed into being a CMO, and we've moved from 1G all the way through to 100 kilo in-house manufacturing of the enzymes we discovered from metagenomics. And for the metagenomics, just as a quick pipeline for the enzyme discovery that we run by, to simplify everything, it obviously starts from an environmental sample, from a sample of choice from a diverse site, or just from the soil outside your own house. With the DNA, we do an extraction, sequence it, analyze it, we select targets, clone, express, screen, and then moving on to how we collaborate with pharma with the free of charge toolkit enzymes. This is the point where we pass that over to our collaborators to screen for their hits, or we screen in-house, we engineer, we scale up. And this is just a basic pipeline how all of our enzymes move through to end up in the hands of pharma. So there are other pipelines from metagenomics that we don't want to talk about today, microfluidics, etc. But moving on, so in 2010, we performed our first environmental sample in the northeast of England, where we took samples from contaminated land to try and identify enzymes for biofuel projects. We didn't perform any sequencing of these samples, and it was just strict molecular biology techniques. We identified a few enzymes, and one got taken forward to the project. It was only until 2014 when the Nagoya Protocol was ratified in the UK, where we were sort of pushed into doing more metagenomic discovery and to do this thing properly. We were unsure at the time on what implications the Nagoya Protocol would have and whether digital sequencing information was going to be included. And even now, we're still not entirely sure how DSI is going to impact moving forwards. So that was one reason, and it's also nice to have your own sequences that aren't found in other public databases. So from 2014 to 2017, we explored a number of sites around the UK, and from this data, we generated around 25 billion base pairs of unique sequence. This has led over the years up to current day of around 2,500 enzymes cloned from this initial sampling effort, where all of the creds in our current proteomics panel, around 600 enzymes are found from these samples, and all of the IREDs, which were found in the IRED work, which was recently published in the NChem paper, these all came from this initial sampling effort. So there's worthy enzymes just from a very, at the time, a boring collection from just soil. So we moved on in 2018 to try and scout for marine metagenomics. It was something we'd never done at Proteomics. We'd never tried to sample seawater. So we set out on a little boat that we hired for a day, just off the coast of Newcastle in the north of England, and we sampled 500 litres of seawater that we concentrated down to just a 10 mil fraction. We then isolated the DNA from the sample, sequenced it using a MiSeq, and then just used sequence-based, nothing clever, to develop a panel of ketoreductases, which I'll go into more detail later, to try and identify cold active enzymes. So in 2019, we have a UK project which is still running in the UK called Newton Baba. And this project allowed us to perform a UK-wide compostome sample, where we visited all the sites, all the cities, all the areas in between the cities around the UK, taking 247 samples. And from this effort here, we've already sequenced around 100 billion base pairs. And when this sequencing, we have sequencing away at the moment, when this all comes back, we should be around 300 billion base pairs from this single effort alone. There's a lot of data here, and we're not even too sure how to analyze it all for sort of scientific reasons, but for enzymes, it's just loaded full of everything we need. In 2020, we had another project which allowed us to try and identify extreme files from the UK. And the UK is a boring place, there's no hot vents, and we struggled to find cool areas to look at. So we found an old steelworks. There was steelworks on this hill at the bottom, and this hill on the left is where they dumped all the waste. This now runs off into this lake on the left-hand side of this bottom picture. And this lake, from one end of the lake, goes from pH 10.5 all the way through to pH 5.6. And we just sampled the soil all the way around the side of this lake, taking various samples, and we sequenced 32 billion base pairs of sequencing from here. There's a number of enzymes in the current toolkits which come from these samples. In 2021, we decided we need to do something really cool in the middle of a pandemic because everyone needs a little break. So we attempted a circumnavigation in the UK. So this circumnavigation was in place to basically take a surface sample to concentrate up on board. We set up a molecular biology lab on board, and we were going to process all the organisms on board and take continuous samples the entire way around the UK at depth and at the surface. We processed on board, we sequenced on board using Oxford Nanopore, and then when we got back onto the land, we sent this away for deep sequencing using Novasec. And we estimate from this project we were going to get around 600 billion base pairs. This is now looking like we will hit the trillion base pairs just from these sequences alone. And we've got a log on the website at the bottom there. So this project itself, so we were going to continue sample using basically a piece of scaffolding pipe off the side of the ship, just down into the surface at 2 meters depth, or 0 when the boat tips sideways. And we also had deep sequencing, deep sampling, which was a pipe that went down to 30 meters. And at one point in the sampling trip, we took a sample at 100 meters. So these were pumped on board to a pump station where we held around a ton of water on board at any one time. They were concentrated using filtration down to 150 ml sample, which was then taken further and isolated and extracted on board using this really posh looking molecular biology lab, which was underneath the front of the boat. And then we wanted to make sure that on day one, we weren't sequencing just one bacteria just over and over and over. So we wanted to make sure the samples were diverse. So we had a bioinformatician on board who was happy in his little lab at the front. This was Professor Neil Whippat from Newcastle University. He came on the trip with us. We worked with Neil for years now, and he's a very good bioinformatician. So we knew the project was working from day one. So the plan was to get on the boat in Macduff and then travel over 14 days all the way around the UK. So this seemed like a great idea on the first few days when it was lovely weather. We were sat on the side of the boat, just having a cup of tea and a bacon sandwich. And we tested all the equipment. We obtained our first bacterial parts. We extracted it, prepped it, the first sequencing reads, and everyone was happy. And we were told that it wasn't going to be like this. It's February, you're being silly, the weather's going to be awful. So we went round and hit the Atlantic. And as soon as we hit the Atlantic, we got a true taste of how bad the weather would be. While this was happening, I was sat up in the top here doing a grant review meeting, which was almost impossible. And at the front of the boat was this area here. You can see the lab was still running, the nanopore was still sequencing. Some of the students we had with us were in here just sort of clinging on for dear life. During the storm, we had a fire in the lab. It was an absolute nightmare. And we were told, as you can see in the top right picture, this was a wind radar at the time for where we got hit. And we were told if the map goes yellow, it's going to be really rough. And they didn't say anything about gale force 11. And it was quite an experience. But we pushed on. And and people always say, why winter? And it was to avoid the blooms in the sea that happened from March onwards. And it's almost impossible to get a nice bacterial sample without contaminating it full of single organisms. So we moved on from there. We headed south, ready to go past the strait between Ireland and Scotland. And again, we were just hit by bad weather after bad weather after bad weather. We were getting quite sick of it. We weren't allowed to leave the boat because of COVID. So we had to sleep on the boat, stay on the boat over 2 weeks. And it got to the point where we had to turn around here because it was impossible to get through this strait here towards the Isle of Man. So then the project took a turn. At the point of turning, we had taken 22 different samples, all diverse. We were sequencing all the way around to check they were different. And we returned north from here. So we spent 14 days at sea. We collected about 25 tons of seawater, which were processed down to 50 mil aliquots. 23 samples at the surface, 22 at 30 meter depth, one sample at 100 meter depth, which was taken above the deepest point in the UK where they test all the military submarines. We performed the microplastic survey with the SAMS Institute in Oban, and we've sequenced around 300 billion base specimens already, with around 600 billion currently being processed. And from each initial analysis, each sample seems to contain roughly between 6,000 and 8,000 creds, 1,000 direds, a couple of thousand transaminases, as you would expect from that level of organisms. And we had one laboratory fire and a broken centrifuge. And these enzymes are already being prototyped by pharma, probably by some people listening to this talk that don't even realize that enzymes came from these samples. But yes, so what we do when we've got the samples and the fun's over, we have our in-house Bioinformatics Platform, which allows us to curate the databases, analyze the databases quickly, uses automatic maximum diversity selection with conserved sequence motifs, etc. All standard stuff now. And then we synthesize, clone, express, and then we supply these materials free of charge to pharma in vials and plates. A lot of people now know this is our standard practice at Prosomics, and it works really well. And this is just an example of the cold active enzymes that I mentioned from the first North Sea sample. So we took these samples from the North Sea. There was around, I think, 1,200 creds in the whole sample. And using the platform, the Bioinformatics Platform, it selected 96 enzymes that it believed were either the most diverse or the most likely to be psychrophilic and cold active in that set. So we took all these red dots and the predicted psychrophilic ones being the purple dots. And our aim was to identify a cold active enzyme, but also one, maybe a cold active enzyme that could survive the cold, but also be active at 30, 40, 50 degrees. And that's what we wanted to achieve there. So these were also of interest here because this was a sequence-based similarity with a 30% cutoff. So we know that every gene that shares more than one node is more than 30%. So these single nodes down the bottom here, we were quite interested in see if we had any activity from these being so dissimilar to any other enzyme in the panel. So we used our in-house cred to go method that we've now characterized hundreds of creds against hundreds of substrates. It's very standard. We offer this also free of charge to customers, and we characterized the new panel with one indinol and other substrates, such as an example substrate, but we characterized one indinol at different temperatures, running the assay at different temperatures. And we saw a few enzymes that preferred 10 degrees over 25. Most of the enzymes were happy at 10 or 25. And this was just plotting where the hits are that are identified to show that some of these diverse ones with no known similar enzymes still had retained activity. It was quite useful. So we then thought, why not try and push the temperature of the assay up? We weren't too sure how the stability of the assay would work, but it works quite well at 45. At 55, it starts to precipitate. But we did identify a number of hits which would retain activity through the temperatures. Number 45 was a control, so the bottom well H8 was a control in these two temperatures. So this was a thermos cred, and we saw some good results. So we realized a number of enzymes retain activity at 10, with some retaining at 55. We identified all these enzymes from the North Sea sample where they're in seawater, which doesn't vary much from 10 degrees all year round. And 2 creds from this plate, which we released 2 years ago, have already made it now into manufacturing at large scale. And moving on to a case study about why we really need these large data sets when there's obviously public domains, but we like to try and do everything without touching any public data because we just don't know if the Nagoya is going to happen or if digital sequence information get taken out. So we'd rather do everything ourselves and do everything in-house. So we got approached by a customer who had an enzyme that was already in the process where they were looking to improve activity and improve stability if possible. If it didn't happen, they weren't too bothered. So it was a nice, easy project. So they first started, they screened our transaminase panel. They identified within a week that a few of the enzymes were 2 to 8 times better than their current enzyme they had, which was a great result to start with. And then we moved on and we said, we'll develop you a quick metagenomic homolog panel based on the hits. They screened it, analyzed it, and again, we found a better enzyme than our original better enzyme, which isn't a surprise when you look at the numbers of enzymes. But whilst doing this, we used our large data set of transaminases and used this to work out what nature did best at every single position that was involved in the active site of our hits versus the original customer hit, and to re-evolve back towards what nature did from what we had. And we'd never really tested this before, but it worked, and we had 3 mutations which identified with massively significant improvement. So we then took both of these together, combined them, and we engineered the new hit with the mutations we'd identified, and it improved on the original toolkit enzyme 6 times over our new hit from here, which in total was a 40 times improvement on activity from the original customer's enzyme. But whilst doing all these steps, we'd accidentally increased the stability of the enzyme to go from 5 recycles in a mobilized form to more than 20, where they were testing it at 20 times, 25 times recycles, and it was just losing less than 1% activity every cycle. So that enzyme now is going to multi-ton manufacture, and it's quite exciting. And just to finish off, the impact these enzymes are having, because it's not just in pharma, they're having an impact in academic circles as well. So since 2014, over 600 billion base pairs were sequenced. We've got more than 4,000 enzymes now that are cloned from UK environmental samples. They're being utilised in a number of UK and EU projects now. So there's a number of enzymes that are identified for pharma applications. A large number have been scaled for manufacture, and there's again a number of these now which have had patents filed by our customers, not by Prosomics. And a number of these enzymes, as people will know, have led to high impact publications. And these are ultimately from close collaborations with academic groups. We don't seek to publish at Prosomics ourselves, but we do seek to collaborate. So we always say new collaborations are welcome, and we look forward to speaking to new people. And 2 specific families. So look at the creds. We've had multiple multi-kg manufacturing events from the creds. There's 7 running at kilo scale, a number of customer patents, loads of publications. And with the IREDs, it's a similar story. We've had multiple multi-kg manufacturing events, including scaling one of these enzymes from the metagenomics to 100 kg manufacture. We've had a large number of numerous early stage hits for new pharma projects over the last 2 years. There's been a large number of publications from our really close collaboration with Nick Turner's lab, including Angwanti, Jax, and many, many more. And that also led on to the Raw Site Chemistry Prize with Nick Turner's lab and Glaxo and York. That was a really nice top up during the pandemic for us here. It was really good. And I'd just like to acknowledge some people. So I'd like to acknowledge Intelligence. So Intelligence are a small spin-out company in Newcastle. They've helped us with all our metagenomic data analysis, data assembly. And if you ever need anything to do with metagenomics, bioinformatics, speak to these guys. They're really, really good, and they'll be happy to help. So, and then obviously I would like to thank the Turner Group, because about their collaboration, we wouldn't know what half our enzymes do. They're brilliant night down there. And Whipat Group, from all what was mentioned earlier, and for the rest of the guys here, including Bob on the boat, who kept us alive in the storm. Thank you.
Great. Okay. Thank you for the great talk. I'm going to pass this over to Anya now, who will field the questions.
Thank you so much, James, for taking on this kind of wavy journey. I feel like we should start digging in our backyards, and our job description is probably not as exciting as yours. As the questions are pouring in, I wanted to briefly ask you about soluble protein expression in E. coli, because obviously you're very diverse fragments that you identified, and what's the kind of average success rate with functional proteins, or is it actually not something that one can easily determine?
Yeah, sorry, I missed the middle bit of that section there of the question, but I think you were just asking how we see what level of solubility we see from environmental samples. We did an initial metagenomics back in, was the 2014 version, where we looked at taking it directly PCRing from the environmental sample itself and working out using the native enzymes rather than the native DNA rather than the codon optimized DNA. We saw incredibly good soluble expression to say there were native enzymes. And I think it's CRED 100 that refers to about CRED 300 in our panel. They're all PCR clones, so they're all the native DNA. Whereas when, obviously in 2015, 2016, people like Twist Bioscience came along and the price of DNA synthesis fell, we just then moved to codon optimizing everything for our expression anyway. So we don't worry too much about sort of the problems with the organism with expression. But sometimes we have trouble, depending on the enzyme class, obviously. You look at things like GoAzers, you're never going to have fun. But some things are easy, and stuff we work with all the time, like IREDs and CREDs, even if they're from fungal strains, we seem to express these quite well in our systems.
Excellent. So there's a couple of questions online. I gave the speaking rights to Anna Toplag from Netherlands. Anna, would you like to ask your question live? If not, maybe I can read it out. So Anna is asking, what was the time frame of the project from a transaminase screen to multi-ton production?
Yeah, so we weren't involved with the final production because we sold the rights to that specific modified enzyme along. But from taking the project on to allowing from the initial talk through the screening, the mutagenesis, and the homolog selection, we were only looking at 3 to 4 months there, and then the products got acquired at the end of that time before we got to sort of scale it in-house.
Okay, thank you. And Mate, do you want to ask your question live?
Yeah, I think if you can hear me.
Yeah.
James, fantastic talk. I have a question because you slided a bit about the way you were assessing the Kyrates. So they were screening the activity. Can you slightly elaborate about the way you're actually using this colorimetric system for checking out quickly thousands of your proteins? This were interesting.
There we are. So there's the equation.
Okay.
So that's probably the easiest way to explain it, to show it. I'm not the person who developed this, and Simon will be able to talk more about it, but it's as simple as this. So it's working in the reverse direction from typically, and it's just a 94 enzyme based dye assay using a diaphrase to drive it forwards. It's the same principle which we use for our IRED to go as well as credit to go here.
Okay, thanks a lot.
Okay.
Hi, James.
Okay.
I have a question.
Yeah, Yang, is that you?
Yeah. Hi, Anya. Great to see you here.
Good to see you. I think you have a question that you can ask on behalf of many people online today, so please go ahead.
Oh, yeah. So, James, I have a quick question. So, my name is Yang, assistant professor at UCSB. We have a very specific question about your proteomics enzyme kits. So these kits are wonderful tools for us and for many other researchers in the field of organic synthesis. So I wonder if we like to purchase some of these kits, which ones do we start, and how many reactions can we screen with the kits you provide?
Yeah, we don't sell kits to anyone really. So we work collaboratively with pharma, with academics, and you'll never really find anyone that's bought a whole kit from us. So we'd be happy to collaborate, and we do this in a very collaborative fashion where we'll give you enzymes in plates, and you report results to us, and it helps us build our kits here in-house because it allows us we know where the active enzymes are. And this is how we've worked well with Nick's group over the years, where they say, oh, wow, this iRed's really good. And then we just build a panel of 50 iReds based on the good iReds, and it allows us to iteratively build them almost without having to screen them in depth in-house here. So we work quite collaboratively with a lot of academic groups now. And it does help us.
That's wonderful.
We'd be happy to collaborate, and it works well as well, where the academic groups then sort of collaborate backwards with their own enzymes that they've discovered, and we commercialize them on behalf of the academic groups and provide royalties back to the academic group themselves, which works well, and a large number of groups are doing that with us at the moment.
Great, great. Yeah, can we invite you to give a seminar to UCSB? I'll be in touch with you. I emailed you in the over the past couple of days, but just wanted to speak with you during this virtual meeting. So hopefully we can invite you and bring the expertise of proteomics to the UCSB community.
Yeah, no problem.
So I'll be in touch with you by email then. Thank you. It's great to talk with you here.
Cheers.
Great, great. It's hard to say no in public. But okay, I think we can pause here. And Simon and James, thank you so much for being here and for your talk. There's many more questions online in the chat room, so we should continue the discussion there. And now we should move on to our next presentation. So Andrew, back to you.
Great. Yeah, so our next speaker is going to be Nathan Schmidt from Ginkgo Bioworks, and Fiona Quinn is going to introduce them. So you can go ahead and unmute, share your screen, and you can get started.
Great. Hi, everyone. My name is Fiona, and I'm a protein engineer on the protein engineering team at Ginkgo Bioworks. And it is my great pleasure to be here today and to introduce Dr. Nathan Schmidt, who is a senior protein engineer at Ginkgo, who is going to talk about the very exciting enzyme and protein engineering work we are doing at Ginkgo. Nate received his PhD in physics from the University of Illinois at Urbana-Champaign under the direction of Professor Gerard Wang, where he focused on the interactions of antimicrobial and cell-penetrating peptides with biological membranes using many biophysical tools such as small-angle X-ray light scattering, confocal microscopy, and bioinformatics. Following his PhD, Nate continued his work in Dr. Wang's lab at UCLA and designed membrane-disrupting peptides to design multifunctional antibiotics. In 2014, Nate became a postdoc in Professor Bill DeGratto's lab at UCSF, where he studied the membrane protein histidine kinases and conducted de novo design of multi-domain helical bundle metalloenzymes using X-ray crystallography, experimental enzymatic and biophysical assays, and computational protein tools. Nate then joined Ginkgo in 2018 with his wealth of expertise, and so please join me in welcoming Dr. Nathan Schmidt.
Great. Well, hello, everybody. Happy to be here today. I want to thank our hosts for inviting the Ginkgo protein engineering team to tell you all more about what we do. So yeah, like Fiona mentioned, I started at Ginkgo in 2018, so I've been here for a little bit over 3 years. And when I started at Ginkgo, protein engineering was a very new team. And so over the past 3 years or so, it's been really fun to work with other fellow protein engineers in building out a lot of the capabilities that we use to optimize and design proteins and enzymes, and to offer these services to Ginkgo as a whole. So just to give you an introduction to Ginkgo Bioworks as a whole, this is it really by the numbers. The company was founded in 2008. Earlier this year, we actually went public, and so this was a very exciting thing for a number of different reasons. But one of the really great parts about this is that we now have, this ability to really scale out our platform to show everybody, what the way that Ginkgo looks at organism engineering, that how well it can really work for a variety of different applications. Now, in terms of the size of the company, we're just over 650 people at this point. In terms of robots, we're somewhere around a little bit over 300, and that kind of gives you a picture about the way that Ginkgo thinks about doing science, and that when at all possible, we try to leverage, robotics and automation to ultimately achieve our goal, which is to make biology easier to engineer. And I'll sort of explain to you why we look at our job in this sort of context as opposed to a more product-driven one. In terms of size, at this point, we have over 200,000 square feet in the greater Boston area, split between the Seaport area in Cambridge. We also have a space in Northern California, in the Bay Area, and then recently we acquired Dutch DNA, and so now we have Ginkgo, I guess. We have Ginkgo folks in the Netherlands as well. In terms of like why Ginkgo looks at our job as making biology easier to engineer, it's important to sort of distinguish that Ginkgo doesn't typically develop its own products. What we instead do is we partner with others that want to design organisms for a specific purpose, and we use our Foundry platform to help them to create these organisms as specified in a very quick and automated fashion. And so this requires that we use cutting edge developments in DNA synthesis, robotics, analytics, fast iteration through design test build cycles, design build test cycles. And then since we are routinely working with a large amount of data and we are generating large amounts of data, we also incorporate machine learning into our workflows to inform the next steps in the various projects. The partners that we work with, this is sort of splitting up based upon on the top ones within the pharmaceutical space, and then on the bottom ones that are in non-therapeutics, but you can see that there's really a wide range of people that we partner with to offer our platform to ultimately, design organisms to meet their specifications and to do this quickly and efficiently. In terms of how this Ginkgo platform works, you can roughly split it into two different portions. There's the Ginkgo Foundry, and the foundries you can think about as basically being a very large factory for doing high throughput design, build, and testing of various organisms. The other side of it is Ginkgo Codebase. Codebase, I would phrase it in terms of just information that's out there. And so information in terms of genetic code, we use public and proprietary databases when we design our organisms. We also have organism database or organism banks as well. And so the whole idea behind this is to turn synthetic biology and bioengineering into something that looks a lot like software development. And we know that's an oversimplification, but you sort of have to start somewhere. And so the idea here is a partner comes with us with certain specifications for an organism, and then we use our factory and the foundry and our code base in order to design an organism to meet these specifications. The Foundry itself is really kind of split up into different teams. We operate collectively in terms of a design, build, test cycle. The protein engineering department, we are mostly corralled within the design portion of this. And so the typical request that we will get is someone will come to us with a protein that doesn't work very well for whatever reason. It could be that the kinetic parameters are not suitable and it's a bottleneck in a pathway. It could be that it doesn't express very well. And so what our job is to do is to use that protein and learn and from it design protein engineering libraries that should be able to solve the issues that the team is having with the typical enzyme. And so we generate these protein engineering libraries. They're then synthesized through our build team. The DNA is then transformed into organisms, and then they're ported over to our test team, who can screen this in ultra high throughput fashion. And at this point, we can once we get to test, we have the data, and if the results are suitable, we can then move them on to doing fermentation and help our customers to do scaling up and scale down and process deployment. And if we need to do another round of DBT, then we can use the information that we learned from the activity measurements of the original library and go ahead and repeat the process. Codebase is, as I said before, it's really just big data in terms of the number of, both public and proprietary databases that we use when we go ahead and design organisms. And then we also, of course, also have strain banks. And then in certain cases, we will also work with or actually acquire other companies for various strains of interest that we would like to onboard and build out. So, focusing on protein engineering at Ginkgo Bioworks, I think you can see that due to sort of the diversity of programs that Ginkgo works at, the protein engineering team, has to really sort of think about, what are the most important things to build out to support these different programs, and how to have offerings that will allow us to support very diverse, scientific projects. This is the team. Right now we have 9 people in total. We're led by Emily Renbeck. She's the head of protein engineering, and our team in general is I would say roughly split, at least historically, 50-50 between wet lab and computational protein engineering. And really, our job is really dedicated to improving proteins expression in the case of enzymes, kinetic parameters, and so on. Now, we do this using really a multifaceted approach because we work on a lot of different projects. Although eventually you begin to see some, sort of kind of collective trends between the different projects. But at any rate, because we work on a lot of projects, we have developed, sort of a suite of different ways to approach protein engineering. On the left-hand side, we do computational design. So in terms of structure-based design, this is Rosetta Schrodinger, and then we have sequence-based design tools that we use as well. Smart library design is basically an iterative process to protein engineering where you follow a design, build, test, learn cycle, where you design a library, you test it, and then based upon the activity, then you can go ahead and use the learnings there to design the next round until you complete, the task of building an enzyme that's suitable for specifications. Then finally, we have an experimental wing that relies or that is mostly high throughput. So these are going to be screens where we handle mostly things where you can relate a protein phenotype to some type of fluorescent signal. And so typically these libraries that we're screening, they're using, FACS-based selection tools coupled with NGS. And so the libraries that we're looking at here are hundreds of thousands to millions. But we really combine, a suite of both computational and experimental tools to help design proteins. Starting out with smart library design, really what I'm saying here is using machine learning. And I have a single case study here to talk about one of the ways in which we deploy machine learning as part of our workflows. So in this situation, we had a terpene synthase that had very poor product promiscuity. And so this was causing a bottleneck in the pathway because we were losing carbon to unwanted products. And so it really became an issue of how to engineer product specificity into this enzyme. And the solution here was really to apply an iterative design workflow for this. And so what we had at our disposal at this point was we had our high throughput screening team had developed these assays where we could read out 5 different products that the terpene synthase was producing. And we also had a few thousand variants. So these are going to be variants in the form of protein mutations to a template sequence that are then linked to their product profiles. From this data set, we were able to train a few different machine learning models to go ahead and learn sequence activity relationships between the variants and the products they produce. And after training the models and choosing the best ones, we then went ahead and had them score a variety of different mutations that were then tested. And so this is the result from this endeavor. You're looking at a at a histogram plot here, where the initial learning libraries are shown in black here. You can see that the percent desired product profile isn't great. We're largely under 50%, whereas the one that was designed using machine learning, we've now really shifted this product profile to largely above 50% of what we want. And then, of course, there are some that are at 80 to almost 90% product specificity. So this is just one way in which we go about engineering proteins to suit program goals. High throughput assays. The one that we typically use the most is when we have situations where proteins just simply don't express well. And in these type of cases, we can use GFP fusion reporter system to design variant libraries with the goal of improving protein abundance. And with that is usually coupled stability. The idea behind this assay is that you fuse together a moderately stable GFP construct with your protein of interest. And you do this for a large variant of libraries, hundreds of thousands to even up to 1 million. If your protein itself is very stable, it will likewise induce GFP to fold into a functional state where it can then fluoresce and give you signal. Whereas if your protein is not stable, then you don't get GFP fluorescence. And so after you create this library, many variants, you can transform it in a one pot transformation, and then use VAX to select the ones with the highest fluorescence. And then take these and use deep sequencing to see what they are and see which sequences are enriched and which sequences are depleted in the ones that have the highest fluorescence. Now, this is not something that Ginkgo developed. Like, we were not by any means the first to develop this, of course, but one thing that we do have is we have the ability to do this in a very standardized fashion. We also can lean on other teams at the company to help us to do things in a more efficient fashion. We have a whole NGS team that can go ahead and do all of the sequencing. And so this really allows us to shorten the amount of time that it takes to generate one of these libraries. And then this is just what you see as a result, which is, of course, along the x-axis, you have all the positions, and then the y is showing you each mutation, and then the color is whether or not it was enriched or depleted in the top. I think this would probably be 5% that were selected. And so this gives you an idea of which positions you might want to focus on for generating mutations that should improve protein stability. Finally, computational design. This is both structure and sequence based. The example that I have for you here was something that Ginkgo did in response to the pandemic. So in spring 2020, when the pandemic really hit the United States rather hard and suddenly, Ginkgo decided to increase our in-house capacity for antigen variant design. And so the one that we selected was the one that was the most relevant to do so, which was the spike protein of SARS-CoV-2. And what we ended up doing was we took the protein and created a variant library with the goal of trying to improve the solubility of this protein, the stability of the protein, ideally what conformational states it selects, with the goal that certain ones are more relevant for antibodies than others. And so at the end of the day, this became like really kind of I would say a multi-dimensional protein engineering problem, where we took into account a lot of different, aspects of this protein itself to try to improve protein solubility and stability. I can tell you from working directly with this protein, it's not an easy one to work with. And then at the same time, make sure that we're not we're not destroying any of the epitopes that are known to be important for the actual function of spike protein. So this is the data from the library that we designed. What you're seeing here is this is the template, and then these are the variants. And what you're looking at in this case is the rate of binding, so K on. We looked at three different proteins that are known to bind to spike ECT, ACE2, which of course is the receptor that SARS-CoV-2 uses to get into the cell by binding with it through its spike protein, and then some neutralizing antibodies. And you can see that we were able to improve the K-on rate of binding in this case. Another issue with this protein is it's just really difficult to express. It doesn't express well at all. And so one of the other things that we were hoping to do was to be able to improve the expression of the protein. So if you wanted to scale it up for deployment in diagnostic testing, that it would be easier to express. And in this case, we're also able to improve the yield, several fold over the template. And so with that, I'd just like to thank everybody again for attending this, and tell you that, Ginkgo is also hiring in the protein engineering department and at the company as a whole. And so, please let us know if you're interested.
Okay, great. Thank you for the talk. I think that we can pass this over to Anya for a question.
Yeah, thank you so much, Nathan. I have a couple of philosophical questions. So with 300 robots, clearly screening is not a limitation for you. Could you comment, where do you think the future is? Is it more in design and rational function design, or is it still in screening, or what's your take on that?
So the way that I look at this is the design space of a protein is so ridiculously large that in a lot of ways it's kind of both, right? Having more data is really helpful because we know that the landscapes of protein fitness are ridiculously complex. And so in that sense, you do want to have as much data as you possibly can to try to understand, the effects of mutations on protein function. However, I don't think rational design is necessarily going anywhere because you just can't test everything. You can't even test. I mean, what you can test is so small that I really think that it's kind of the merging of both and coming up with ways to sort of use rational design in such a way where you're trying to do it in a way that is built for larger data set collection. And that's something that was very different from me from when I was in academia, where you're testing 10 proteins and that's all you got,
Okay, and if I may, another question. So in vitro cascades or biosynthetic pathways and whole organisms, where do you think the future is?
Synthetic pathways and whole organisms. Are you talking of like, Ginkgo routinely, creates pathways in organisms that are, 6-7 steps that are, recapitulated in microbes that originally come from plants. I guess I'm sort of like, is it? I mean, we do this already, and I think we're going to continue to do it. I would say it's very complicated to do, and so there's a lot of troubleshooting that goes into it. And so I think getting better sort of metabolic models would be something that's really important for getting better ideas about what's going to work and what's not going to work. Yeah, that's what I would say, but I'm not sure if I actually answered the question.
I think that it's a good answer. I mean, Ginkgo is an organism company. We focus on in vitro cascades outside of the cell because that's unique to pharma industry. But maybe in the future, I will it will be different. Okay, we have more questions online. So Lucy, would you like to ask your question live, please?
Hi, thanks, Anya. Hi, Nathan. Lucian Wong from University of Manchester. Thanks for the talk. Regarding your GFP fusion assays, have you, in your experience, found a correlation between getting high fluorescence and soluble candidates very good? The reason I'm asking is we've tried this assay ourselves, but we find it's pretty hit or miss. We've had candidates with high fluorescence which turn out not to be soluble. And I'm just kind of wondering whether it's maybe we're just simply not screening enough samples. I mean, we're going with libraries of, I guess, 100 candidates, and yeah, we've really not found the assay to be very good. So I'm wondering whether you've got some, I guess, some tips or tricks to handle that.
I know that, like, one of the things that we know is vitally important is it's like anything else that you get what you screen for. So, you know, when using these assays, if we're going to be putting this in E. coli, we obviously screen in E. coli. That's, like, you know, important. I would say that, like, we've had more success than failures with it, but there are cases where we have, I can think of one case in particular where we did do a library design on a protein, and the mutations that we identified for improving stability, they really just didn't translate to how well the protein worked in the cell. This could have also been that it's an enzyme, and sometimes you hurt stability, or you improve stability, you actually hurt catalysis. But yeah, like, I think with any of these things, we look at it as it's something that we can do that is very informative, but there are situations where, you know, it's not going to give you exactly the answer that you're looking for.
Yeah, great. Yeah, exactly. It didn't in our case anyway, but thank you very much.
Mm-hmm. Okay, the next question is from David Ricardo Figueroa. Would you like to ask your question live, David?
Yes, sure. Can you hear me?
Yep.
Thank you, Nathan, for your presentation. I was wondering, I mean, how computational techniques, I mean, molecular dynamics, maybe some reaction mechanism calculations, the hybrid QMM calculation are using in the protein design, like problems that you attach. It's because I asked this because I don't know if maybe in Ginkgo this screening, in vitro screening approaches are preferred over molecular dynamics and machine learning approaches.
So in terms of the computational approaches that we typically use, we don't use any QMMM at this point. It's molecular dynamics, and we use Rosetta as well to do protein engineering. So a lot of times for us, it's really a matter of what data we're going to get back is kind of gives us an idea about what sort of tools that we should use to engineer the protein. And with high throughput screening, you don't always get, you know, the kinetic parameters that you need in order to really make, you know, like really drill down into, you know, mechanism of action. And so like, I found that for the structure-based tools that we typically use, that they're pretty good for the library scales and the sizes that we use. Now in the future, you know, with increases or improvements in microfluidics and high throughput enzyme kinetics, then I could definitely see a situation where a lot of these other techniques that you give you complementary and sometimes better information than what, you know, than what we currently use would have application as well.
Thank you very much.
Okay, I think in the interest of time, we should shift the rest of the questions and discussion to the Q&A box and the chat room. Thank you so much, Nathan, again for your talk, and it was great to see what you guys are doing at Ginkgo. And now back to Andrew again.
Great. Okay, yeah, so we have one more industrial talk today, and it will be by Jesus Seco Moral from Zivmol, and I'll be introduced by Maria. So you guys can unmute and share, and we can start with our final talk. Are you there?
Hello, do you hear me well?
Hi, yes. Is Maria online to introduce you?
I don't know.
I don't know if I see her online.
Let's see if she's in the.
She could be in the just the general attendance. Yeah, let's check real fast.
Okay. I cannot see Maria.
Well, if you want, I can introduce myself.
Yeah, we might have to do that. So if you don't mind giving yourself a brief introduction, and then you can go ahead and go ahead.
Yeah, it's quite a level. Yeah, well, my name is Jesus Seco, so I'm working at Zivmol since 2 years ago, where I basically develop computational methods for doing protein engineering, and also to discover novel enzymes for novel purposes. So my education was chemistry and also biochemistry, and it has been a long path during the last year because I was moving from purely drug discovery to protein engineering a couple of years ago. And basically, it's an exciting journey. And today, I would like just to present all the work that I've been doing at Zivmol since 2 years ago.
Great.
Okay, so let me tell. Do you see the screen?
Not yet. No.
Okay.
There. I see me. Oh. There we go. Okay. That looks great.
Okay, well, so good afternoon, good morning to everyone. First of all, I would like to thank you, all of you and the organizers, to give me the opportunity to stay here today to present our company and our work and how it can assist you to unravel the enzyme space for novel purposes. So Zivmol, it's a 4-year-old company. We are based here in Barcelona, Spain. We basically applied and developed proprietary molecular modeling software to discover and design highly efficient enzymes for industrial applications. In this way, what we do is we actually provide two main validated services. In one hand, we offer the enzyme search, which is an algorithm to find natural enzyme for your particular needs, and that I will further explain you today, and also the in silico design, where we tailor enzymes toward improved properties by computational mutagenesis. Of course, the computational predictions that we do all the time are supported always by the experimental validation done by our wet lab partners that assist us in the expression purification of the enzymes and in the analytical methods. So after 4 years, our team has expanded significantly with experts in different areas as diverse as chemistry, computer science, biotechnology, and even astrophysics. So, I mean, we are welcome any kind of people for our company. So our team has a strong track record in enzyme engineering and discovery with an accumulated experience over multiple international projects with clients worldwide. And well, the very good thing of this multidisciplinary knowledge is that the team has been able to translate this into our technology stack that allow us to study the enzymatic problems by using different layers of complexity. In some cases, we can easily study the problem by using traditional bioinformatics approaches, like this one, for instance, by using multiple sequence alignment. But as you can imagine, it happens not too much frequent, less than 1% of the projects that we have. So in other projects, what we really need is to convert the enzyme sequence into a three-dimensional representation. Moreover, we need to take into account the flexibility of the enzyme because, as you know, they are not static biological entities. For that reason, we introduce other elements like side chain, backbone flexibility, also the role of the explicit solvent that we are using in the assay, if it is pure water, if we are working with an organic crowded environment. So, and in some cases, what we need to do is to go beyond, and we need to elucidate the quantum at the quantum level, so the mechanism of action, to totally characterize the enzyme that we are going to study. What we are is we are strong supporters of the dogma that computers are the microscope that allow us to access to the atomic wall and help us to generate rational structural hypotheses to fully characterize and understand enzymes. And that's what we really want at the very end of every project. So if we can understand the enzymes, we can just better accelerate the chemical reaction. And with all these computational technologies, we can understand how an enzyme is able to not only lower in the activation energy, but also how we can exploit that information, how we can exploit that kind of enzymes for our industrial applications. And this is crucial because, so enzymes are used more and more in industrial context for the synthesis of products that are not accessible by chemical synthesis, but also to lower infrastructure and operative costs, to use alternative raw materials, and to improve eco-efficiency. Unfortunately, when we take a look to the number of enzyme applications and we compare that to the number of known enzymes that are reported in nature, we see that this number is pretty disbalanced. So we know that we need to understand much more the enzymes in order to exploit them for an industrial context. And well, this is because enzymes have naturally evolved during millions of years, have been shaped by this evolution to make a specific chemical reactions to a specific substrate. And even if you are working with a very similar substrate to the one that is reported in the literature, probably it is not applicable to your industrial process. Fortunately, today we have the methods to partially reproduce from an in silico way the evolution by means of what people call enzyme engineering. In terms of enzyme engineering, we have two main blocks: directed evolution and rational design. While directed evolution has been the option of choice since only requires a minimal knowledge of the system, the rational design relies on the atomistic knowledge obtained through simulations to select a few protein sequence mutants for experimental validation. Apart from that, what we can do is just to take the best of each one of these and use what we call semi-rational approaches that use rational but also random tools to efficiently explore a great part of the enzyme sequence space in a knowledge-driven way. So, and here is where Symbol in silico design technology is allocated. A couple of years ago, what we did was we created the Symbolver that could be described as an in silico multi-approach that combines physics-based molecular modeling, network theory, and machine learning to screen efficiently the enzymatic sequence space, and also to identify the most beneficial mutations for a certain enzyme property in a short period of time. However, today I'm here to talk about how we can perform in silico design, because it's something that many people is doing, how we can do protein engineering, because I would like to go backwards in the enzyme engineering process. And let's talk about the problem. What happens if we don't have a candidate to be used as a starting point for our reaction? So what happens if we don't have any enzyme here with some activity, or if we do know an enzyme, but it's not expressible in our host organism, or it's IP protected and we cannot use that? So what normally happens is that these projects are basically left behind, or probably the project can be done in an enzyme discovery campaign, but it's done by using metagenomics, for instance, but which at the very end, it's really, really expensive. So a couple of years ago, so we noticed that many clients came to us asking about this specific problem. And that's the reason what we did is to develop this software that we call Biomatchmaker that allow us to rapidly search for the best enzymes for a particular reaction among the known natural enzyme space. Briefly, Biomatchmaker combines bioinformatics and 3D molecular modeling to identify candidates for a decided reaction, even when non-natural enzymes are known for that particular process. This is a more detailed diagram of what Biomatchmaker does. So imagine in this case, this particular reaction. Imagine that we want to transform this alcohol to this ketone, but only one of the enantiomers wants to be transformed, the other are not. So what we usually do is we talk with the client, we define the KPIs and other critical parameters that are going to be tested afterwards experimentally, and we introduce that data into our algorithm. And once it is done, so we can start running the algorithm. So the Biomatchmaker automatically scrutinizes public databases to gather sequence data. So these millions of enzyme entries are afterwards clusterized to reduce problem dimensionality. And in some cases, so if we are lucky, what we can do is get access to crystallographic information for these enzymes, but this is not really frequent. So what we have to do in most of the cases is to use traditional homology modeling techniques or advanced artificial intelligence methods, such as the one reported by the people of the AlphaFold, to have a three-dimensional representation of, for our enzyme system. These methods, these models and these crystallographic structures are subjected to quality inspection, but also to conformational exploration to yield an ensemble of enzyme conformations that are going to be analyzed later by molecular docking, where we actually use molecular docking, where apply a hierarchical strategy to save some time. And at the very end, what we get is a ranking of the best enzyme candidates that are ranked accordingly to geometric and energetic parameters. And finally, what we get is what we call a hit enzyme, or a potential hit enzyme for one reaction that we want to test. So actually what we do is we propose several enzymes. It depends. It's something that we agree with the client. This number could be between 10 to 50. And well, it's the client at the very end, or ourselves, that we do the experimental evaluation of this experimental reaction. So let me show you what we did internally here at Trimble in order to validate this tool. So we were at the very beginning looking for enzymes able to perform 5 novel conversions. As you can see here, the substrates that we were analyzing were pretty different each other. I mean, the similarity between them were between 10 to 80%. They were enough descriptive of the different substrates that people from pharma and chemistry companies were talking to us. So this was more or less, let's say, a decoy molecules or decoy substrate to be tested. And what we did in this moment was to test how Biomatchmaker was able to identify new enzymes for this reaction. Just to mention that no enzymes with activity for any of these substrates was reported previously to that. So the Biomatchmaker was able to propose, after evaluating more than 100,000 in silico alcohol dehydrogenases, a set of 16 non-redundant enzymes. As you can see here, we have the heat map similarity between them, so the identity is pretty low between them. So this is something that we always preferentially do, because we try to expand the IP potential probability to identify a hit enzyme in the case you have something, so it is better to have something different in each one of the cases. So actually, the project, from what we started the computational evaluation, we did the experimental evaluation, we purified the enzymes, we did the analytical method. It took around 3 months. We proposed, as I was saying before, around 16 enzymes. Only 10 were finally expressed. For the others were problems. But the very good thing is that from these 10, 6 enzymes reported activity. And the very good thing is that 2 enzymes of these 6, the number 5 and the number 7, were highly active on 5 substrates. So sorry, here you have the activity table. As you can see here, we have the enzyme number 6 that has been selected for further engineering to improve the activity towards these transformations in a joint effort with the people of Aminoverse. In this way, all the positions of this enzyme has been changed by gene saturation mutagenesis, generating a big amount of useful data. And well, the very good thing is all this generated data is now being used to train our machine learning algorithms to find efficient variants for these biotransformations in an alternative approach that we do for the Biomatchmaker. So altogether, these algorithms, along with the ready-to-use enzyme variants, provide a fast and reliable way of enzyme screening that can be used for the projects. So to check for additional capabilities of this software, the Biomatchmaker, we conducted a second validation process that was the continuation of the previous one, but now based on transaminases. So encouraged by the good results that we obtained by alcohol dehydrogenases, we wanted to check if the same algorithm was able to add more value to the chain by investigating other reactions involved in the transformation between the alcohols to amines. In the former enzyme search exploration, we were able to identify 6 candidates. And here, what we wanted to verify if we were able to identify transaminases able to make this final transformation. We continue with the 5 conversions that we were using before, coming from the alcohol dehydrogenases, but here we add 4 additional substrates that were from pharmaceutical relevance. And in total, we were able to propose between 58 to 65 different enzymes, transaminases for this. The very good thing is that for the total number of reactions that we were covering, so we have now experimental activity or experimental data for 6 of them. For these 6, we have activity for 5 of these reaction. So we have for every reaction between 1 to 2 transaminases, and the initial conversions that we are able to obtain is between 10 to 40. So we have been testing this with different amine donors, including IPA. And right now, so we are just trying to obtain information about the remaining 3 reactions for where we are not able to obtain experimental data due to some experimental problems. Sorry. With the positive experience that we obtained with the alcohol dehydrogenases, we encouraged us to develop our own enzyme kit in collaboration with the people of Aminoverse that I'm glad to show you today. These enzyme kits that we call Thymol Aminoverse alcohol dehydrogenases can be used to convert a ketone to aldehydes or alcohol to vice versa for chiral resolution of alcohol racemates, and also for the production of decided chemical alcohol or removal of alcohol. Apart from the natural activity towards multiple alcohol or ketone substrate that could be representative of the pharma, the readout information that we can take from these enzyme kits can be used in conjunction with the machine learning methods and all the platform that we have been developing in order to provide a positive response for you. And in case you want to test it, we are always open to talk with you and to share with you that kind of enzyme kits. And finally, the conclusion of this biomatchmaker. So what we would like to emphasize is this is a promising tool that has not been comparable to any other method previously reported. The algorithm, we can tell you that it's unbiased, so it has been applied to alcohol dehydrogenases, transaminases, but also many other families like nitrilases, esterases, halogenases with good success. So it depends quite a lot. Every project is totally different, but almost in each case, we are able to obtain a hit candidate or a hit active enzyme for the specific target reaction that we are studying. So the very good thing is that we have not only the biomatchmaker, but we also have some other alternative machine learning methodologies that are supporting the biomatchmaker in order to implement this information with our enzyme kits in order to have a better solution in case the enzyme kit that you can test doesn't provide you a good transformation. We can re-engineer very quickly and very rapidly all other mutants or other variants for your reaction with no cost. And well, this is all. So thanks to all of you. And in case you have any questions, so this is my email.
Thank you so much for the talk. We can now pass to Anya to moderate the questions.
Thank you so much, Jesus, for telling us more about your technology. I was curious whether you can comment a little bit how much prior information or do you need to be able to have effective prediction with your algorithms? Does it have to be a well-characterized enzyme class such as transaminase or ketoreductase, or can you do it on a quite a boutique or unusual enzyme too?
Well, it has changed quite a lot during the last month because, I mean, before that we were using basic homology-based templates, techniques that required from other entries or other enzymes to be crystallized in order to make a good predictions. But we always have alternative solutions. Thanks to, for instance, AlphaFold, we are now generating millions, well, not millions, but thousands of different enzyme homology models that can be used. We are also using ab initio models in order to generate, for those cases where AlphaFold is failing, alternative solutions for that. So what we always say is that we need good structural data in order to make the evaluation. But in most of the cases, we don't proceed to apply the algorithm if we see that the structural information is not good enough.
Thank you.
Quick question. So in data, when you provide these active enzymes to your collaborators for evaluation, in the example with the 5 enzymes and the 5 substrates, there was like several fold change in the activity against a lot of those reactions. What's the percent homology of those 5 enzymes? Are they? I'm assuming they're very closely related. Are they very different? Like,
no, we're totally different. It was something between 20 to 30%. Each enzyme was totally different. Yeah, that's the reason we always try to, at the very end of the selection, we try to prioritize different alternative solutions, because in case, I mean, there is no sense for us to give you 10 solutions that are close enough. each other, because if all them are 80-90%, of course you have more chances to have activity. What we always try to do is cover as much as possible the enzymatic or the enzyme landscape in order to potentially expand the IP.
I see. Great, cool.
All right, we have more questions online. So Maciek, do you want to ask your question live?
Yeah, thanks, Anya. I was really wondering, because these tools you have, they actually look too good to be true. So how you can actually implement the learning-based sequence to predict an enantioselect activity of alcohol dehydrogenase? That seems kind of a miraculous. So could you a bit elaborate, if it's not confidential proprietary technology?
Yeah, unfortunately, I'm not the right person for the protein engineering, because my major strong is the biomatchmaker. I mean, I know people in the company are doing good efforts in trying to develop new artificial intelligence based on natural language processing, also based on machine learning, covering all the sequence space in order to make these kind of predictions. But I cannot tell you something, because probably we're totally wrong. But if you want, you can just try to write me an email, and I will try to give you the best response.
Fantastic. But can you tell us how good this enantioselectivity prediction is at the moment? So, because you said it was okay for 6 substrates.
No, it's pretty nice. I mean, it's not perfect, of course. I mean, there are some cases where we say you are going to obtain this enantiomer, and at the very end you obtain the opposite. But fortunately, this happens less frequent, because we are able to optimize quite a lot the computational workflow. But let's say when we say the enantioselectivity is going to be this or this, in 80-90% of the cases we are right.
Well, that sounds fantastic, actually. Thanks a lot.
Yeah, welcome.
All right. Thank you so much, Jesus. If folks have questions to any of our speakers, please ask them either in the Q&A window or on the chat. And now we can move on to our panel parts of today's meetup. And Andrew, back to you again.
Great. Thanks. Yeah, so I'll ask if James, Nathan, and Jesus can unmute and turn on the cameras. Cool. So I do have a couple questions to go through surrounding how you guys got started in this area, what you think is exciting, what you think will be exciting. But as a little icebreaker, I wanted to give you an unprepared question to get us started. Can you each tell me what your favorite enzyme is? So James, can you start?
Yeah, sure. It's probably P450BM3. I spent a lot of my PhD at Prosomics working on that enzyme and mutants of that enzyme. And I probably produced more of that enzyme itself than any other enzyme I produced at the company. So
good answer. Good answer. Nathan, do you have a favorite protein enzyme?
There's so many, but the one that I actually really like were these were tough proteins, but the histidine kinases that I worked on as a postdoc, because they have such a cool mechanism of signal transduction, involves transmitting signals over long distances using coiled coils. And we came up with this really elegant structural mechanism for how we think they work. And it was really, really fun, because it was really hardcore structure-based design. I had a lot of fun working on the project.
Cool. Jesus?
Well, it's not an enzyme, it's a transcriptional factor that I was working during my PhD. This is the androgen receptor that I was basically working during more than 5-6 years with no ability to obtain something rational, because it's extremely complex to see how tiny mutations on this protein can affect in a very different way physiologically, and that's something I really like. I still continue working with that, but with not too much time.
See, very cool. Okay, so we can get into our questions. So first question, what was the path that led you to industrial biocatalysis and protein engineering, and what was your motivation? So let's go in reverse order and start with Jesus. Do you want to tell us a little bit about that?
Yeah, well, my path was a little bit, well, it's not probably a traditional. I mean, I've been working basically all my career in industry, but mostly related with pharma industry. But a couple of years ago, I was noticing that many of the computational methods that could be applied for drug discovery could be perfectly translated to protein engineering. And it's what I decided just to make that switch, because I believe with that expertise, we can contribute to do something totally new. And I'm pretty proud of that, because I was lucky to be at Simon, where I had the opportunity to exploit all this knowledge in the case for the biomatch maker, and that has been my path. But I always encourage people just to be open to any kind of possibility during the PhD and during the postdoc for any kind of transition.
Great. Cool. Nathan?
I really just followed the science that I found exciting. And it was really just, I wanted to do protein and peptide engineering. I did antimicrobial peptide engineering in grad school. That was sort of my first taste. And then I chose, to move into a postdoc where I could learn more about it. It's challenging, but I think it's really fun and rewarding, because when you see that you've improved a protein, it's like, wow, I've got this, In terms of an industrial career, I really sort of wanted a position that would allow me to do protein engineering. I wanted to kind of get in at the ground floor and work at a company where I thought that we could really sort of build up, a suite of tools to do it, and that would provide us with the resources. A lot of times, you have these great ideas, but if you don't have the resources to do them, then they just stay at ideas. So, that was really what I was looking for.
Yeah, that's a good point. Certainly, there's a wealth of resources available in the industrial side of things.
Yeah.
You get a lot of really cool science that might be inaccessible in an academic sense. I think that's kind of maybe where my motivation came from too.
Yeah.
James?
Yeah, it all started, as Simon mentioned earlier, before I even started at university, I'd take some work experience at Prosomics, and through my first and second years of being an undergraduate, I'd worked at Prosomics through the summers on an internship style time. And during this time, I'd helped develop the initial set of the keto reductase panel that Prosomics now distribute worldwide. So a number of the first 100 enzymes were ones that I worked on before I even had my first degree. And just that time alone, I was seeing these enzymes getting used in pharma and being told they're being used by academics and being published. And it was just seeing before I even graduated that these enzymes that I was developing and helping develop were getting used in processes and basically ultimately helping to save people's lives as you go through the drug discovery phase. And it just, that alone just helped me just justify why I wanted to stay with it and keep on at it, and then spend time looking at P450s and getting frustrated. But it's all worth it in the end. It's been a long route, and as Simon said, I've just gone through the levels at Prosomics now to heading up the team.
Very great. Very cool. Okay. So obviously, this seminar series is kind of designed by nature to allow like academics and industrial professionals to kind of share information and, maybe open channels for collaboration, at least provide a platform to share ideas. Have any of you had any specific academic collaborations that have enabled a technology for you, maybe a direct collaboration or an indirect collaboration? And if so, were there any like barriers that you didn't expect or any difficulties that you had collaborating between academia and industry? So Nathan, I guess we'll start with you.
Yeah, so this is one thing that I think that at Ginkgo we could do a little bit better with, because I actually haven't done much direct collaboration with academia since moving to the industrial side of things. There are ones in discussion at our company, but there's nothing is sort of, I would say, it's not, it doesn't happen as often as I kind of wish it would. I mean, that's not entirely true. Like our Ginkgo's COVID-19 testing, it sprung up as a partnership between Concentric, which is, Ginkgo's testing service for COVID-19, and academic institutions such as Rutgers. And so we do partner with academics and academic labs, but I sort of wish we would do it a little bit more, widespread at the company. But I think this is there are ones in discussion, and so this is a work in progress.
James, how about you?
Yeah, we collaborate quite actively with a large number of academic groups, as everyone's probably aware from the publications that have come out over the last few years. And I'd just have to say that the most impactful one's probably our long-standing collaboration with Nick Turner's group, where they've managed to take some of our panels of enzymes when they're in the early stages and not exactly pushed in the right direction, because just like for the IREDs, for example, we were already working on IREDs in the background, where customers had come to us and said, like, can we start looking at IREDs? And yeah, sure, we'll start putting panels together. And then Nick comes along and says, wow, can we do a panel of 400 IREDs? And we're like, yeah, let's just do it. So we work collaboratively where Nick knows and his group know what's trendy, and they come to us with their ideas, and then we put them ideas into plates and get the plates back to them. They work with the enzymes, and it just works in a nice sort of both groups feeding off each other. They feedback the results, we feedback more enzymes, and we just work the panels up from there to find these great biocatalysts, which in the last 12 months has been a number of publications now, high impact publications for specifically the IREDs, which have been found through this iterative mining that has been fed back over the years. And it's really nice now to see the enzymes being applied in ways we didn't even think were possible.
Any weird challenges with bringing that to full realization?
The only challenge is academics want to publish every sequence. That's the only we like to keep like we're a company, so we like to keep things private, but there's always a trade-off. So
I see. Yep. Jesus, how about you?
Well, nothing too much I can contribute to that, because in my case, I've not been working with academia. I mean, I know some other colleagues from my company that have been working with academic groups in Groningen, here in Barcelona as well. I mean, the basic barrier that we noticed, but it was probably some time ago, not now, is to be aligned in the same project, because in some of the cases from academic people, they have some purposes, they have some goals. From the industry, we have totally opposite goals, and the very good thing is if you can align both to arrive to the same point. That's the only limitation I have seen. But it's always good to collaborate with people, because they open your mind to new ideas, to new possibilities that you have probably no possibility to think about.
Mm-hmm. No, it's definitely a huge factor, is even if the goals and the endpoints are different, finding ways to work together to both achieve that can be sometimes tricky to ride that line. Yeah. Mm-hmm. Cool. Okay. So we'll move into the third question. What developments in the last decade do you think have been most impactful in the field of biocatalysis and protein engineering? So let's start with, I don't know, Nathan, you want to go in first?
Sure. I mean, I would say what I've seen is just big data. Just like protein engineering is so data driven, and with things like advances in DNA synthesis technology and the corresponding decreases in cost, to synthesize DNA, it allows, the generation and screening of these large libraries to get a much better picture of fitness landscapes, NGS to relate, phenotype to activity. And so I think really a lot of it's I see it as big data, like for a lot of different reasons, like protein sequences. I mean, now we're looking at, hundreds of millions in UniProt, way more in proprietary databases, and way more out there. And just that information, it's just so incredible to have at this point. And then protein structures as well, hundreds of thousands of protein structures in the PDB, structural prediction tools that have been published after the last CAPS earlier this year. We're now able to, use a lot of advances in, like MD, computational resources, even the structural revolution, from like cryo-EM to look at, protein complexes. All of these things, we just have so much data now to use to inform protein engineering. So it's a really fun time to work on this.
Yeah, I'd have to agree with you. I have a feeling that the other two responses might be similar in nature, but Jesus, do you have any?
I'm totally absolutely right, and agree with what Nathan say. I would add always the high power computing, because it's incredible to see that with my laptop today I can do what I was doing around 6 or 10 years ago during my PhD, accessing to supercomputing. I mean, it's not comparable, of course, but it's incredible that because today people has a lot of different resources, a lot of power computing process capability, and thanks to that, people can do many things even at their home. So for me, it's incredible to see that. I don't know where we'll arrive in 10 years, let's say, but this in conjunction with, for instance, some advances like the ones done by the people of Baker Group or the people of AlphaFold, that they are pretty close to what the experimental resolution is. It's comparable. I think these have been the greatest thing.
Yeah, absolutely. James, anything to add?
Yeah, just to follow on, it's on the similar lines. Like we're speaking from a small company, without the falling costs of sort of next gen sequencing and synthesis, a lot of the projects we've undertaken over the last 5 years just wouldn't have been possible. I know now, just even comparing 2017 to today, we're paying, I think, something like 64 times less for gene sequencing from metagenomics, and it just means we've got access to so much more data on, which for small companies sometimes tight budgets, and we have to make sure we spend money wise, especially when we're trying to scale up our production side. So just the access to these technologies, and then the miniaturization of these technologies with technologies such as like the Oxford Nanopore system, bringing the sequencing in house. It's just great to see it happening. It means it's available for everyone, and every lab could be sequencing just with minimal budgets with some of the technology we have available now. It's really good.
Absolutely. Yeah. It's exciting to see all that technology become more and more accessible to more and more scientists. So very cool. Yeah. So flip side of that other question, what are you most excited to see in the next decade? Or well, maybe if you had like a wish list of things that you wanted in the next 10 years, what would be the most exciting development you could imagine in protein engineering? Let's start with James. Let's go back to you. Why not?
Yeah. So again, it follows on nicely, and just the continuing fall of DNA synthesis, really, and sort of the miniaturization of that to bring it in house. I know there's a lot of companies working on this to have in-house DNA synthesis, and it's going to be a reality for even small labs quite soon, hopefully. And you couple this with technologies such as incredibly large data sets, AlphaFold, and people have got a route there to find these novel biocatalysts in a very short space of time. And it's going to be quite an exciting few years now.
Yep, cool. Jesus, anything to add?
Yeah, what I would like to see it's in how the artificial intelligence is going to assist us in the process, because I would like to have, I don't know, in 10 years, an assistant that I will tell, do this kind of software and do everything for me. It would be ideal. Because as programmer, I spend so much time doing this, not doing science. And that's something I believe if we can have these kind of tools, I mean, we will do a lot of progress, because we will be able to test many different things. We will be able to do every cycle of iteration of optimization very quickly. And that's something I would like to see in the next years.
Absolutely. Cool. Nathan, anything?
Yeah, I like overall, what the two other people said, I largely I agree with as well. I would just say like technologies that allow us to really scale down experiments while at the same time like scale up the number of samples that we can measure in an experiment. Microfluidic technologies like right now at Ginkgo we use robotics to do everything, and these robots are they're still extremely useful and everything, but they're big, and getting a corresponding the same data that you can do with with some of the things that people have been publishing earlier this year with microfluidics and enzyme kinetic characterization is just remarkable. You save time, you save money, and it's just amazing. And then I would also say just how we're going to use all the data that's generated and use things like machine learning to try to really understand and interpret it and then use it. And particularly, I think it's really exciting period of time to do structure-based design, because we're finally getting enough structural information to really do it well. And like Jesus said with computational power as well. You can now actually model these systems, in a time where you don't have to wait months to get the result. And so this is great.
Yeah, I'll just echo, Merck is also very interested in the miniaturization of the reactions and the maximization of the number of reactions that we can do quickly. So yeah, microfluidics developments in the future will definitely be hugely enabling for protein engineering and biocatalysis. Yeah. Okay, and then I guess we'll move to the final question. Do you guys have any advice for academics or PhD students trying to break into this industry? So Jesus, let's start with you.
Oof. Tough question. I mean, the only advice I could give is to be totally open-minded, because people think when you are in academia, you are doing the good science. When you are in the industry, you are in the evil side of the science, and that's not true. I mean, in industry, you can do many things. So the only advice is go ahead, ask people, talk with people, try to do interaction with other people that are working in industry. Try to work, if you can, some months in order to see if you like it. If you don't like it, okay, no, nothing happens. But I mean, not focus only in academia, because I believe here, at least in Spain, it's quite dramatic to see people mostly devoted to academia, and they are thinking, because if they move to industry, it's some kind of waste of time, because they did a PhD for doing science in the academia, and it's not, for me, the good path.
Cool. Nathan, any advice for academics or PhD students?
I mean, just to sort of start out with, I don't think that you need to break in. We really need you. We really need scientists. There's, oh my gosh, if we had so many more scientists, what would we do? Yeah, and so I wouldn't worry about thinking that it's going to be hard to make the transition. It will take a little bit of time. The timescales in industry are quicker. You have defined milestones and things like that, but you can still do amazing science in industry as well, and you can do it in a way that helps to drive projects forward for the company, and so that you can learn really interesting things and answer some questions that you have as a scientist. And so I mean, more than anything, I would just say, read about different companies, pay close attention to the parts that that you're interested in about them, and and then go after companies that are doing things that really interest you in terms of science, or if you want to go into business development, then look at companies that are focused on this. It's that's, I guess, basically it.
Cool. James?
Yeah, just collaborate as much as you can. We get contacted often from PhD students wanting to either have an internship, a couple of weeks, or just out of their academic environment into a lab, and we'll always support that. We try our best to get these moving forwards, and for the same reason, we try and engage with academics as much as possible, where they can sort of get a feel of what it is like to be in industry, because it is a total different kettle of fish. It's tough, but don't be scared to ask people. And we've had a long history now of collaborating with academics who've wanted to spin out a company to do some testings, and we'll always happily support them and try and give them advice or enzymes or anything that we can help them with. We'll always try and help. So just never be scared to ask someone in industry, because they'll always be there to try and help you out.
Great.
Yeah.
So we have one more question from the chat that I'd like to ask. So this was, do you guys have any opportunities for small internships for PhD students, or know of other companies that might have any sort of training programs related to protein design? So do any of you personally have, I don't know, maybe active internship programs or spaces for PhD students to come in and rotate or something like that? Leave it up for anyone who wants.
In our case, I can say that we are continuously looking for PhD students to engage our company, and for doing internships. I mean, it's a good win-win collaboration process. So if someone wants to contact me, so a pleasure to help.
James and Nathan, any active internship programs?
Yeah, we'll always, if people need experience, they want to do anything, they'll always take them on. The company has a furnished flat nearby the lab that we have for this purpose, where we let people come and just work for a few months or a few weeks, or just to gain some experience, whatever they want to do. And we just put them up in the house and they've got somewhere to live, because it's a bit awkward from where we're located. It's not the most convenient place to get to.
Yeah, we have at Ginkgo a Padawan program, which I really don't like that name, but the idea behind it is that you come and work at Ginkgo for like a year or so. But I think there's additional flexibility with internships as well. Like we have considered doing internships in protein engineering like over a period of a summer or so. And so there's nothing formal yet, but if you contact me and I can put my email in the chat, then please just reach out.
Yeah, I have to say my experience with graduate internships was really, really good. It helped to really solidify my interest in wanting to go into industry. And I spent like a summer at Genentech in 2017. And if you have the opportunity to do an internship in your PhD or even earlier, I highly recommend. So if there are any PhD students out there listening, I would suggest contacting some of these guys and expressing interest. But
likewise, if I can add from a Max perspective, we have internships for undergraduate students. We find that the graduate students, usually the advisors, they want them to have a break in the research intensive PhDs. So we have internships for undergraduates, and because of the competition and the millennial students seem to be applying for those roles very, very early. So we recruit a year ahead for the interns for the following summer.
Yep. Great. So I think that concludes our Q&A panel. So I guess I'll just wrap this up once more by plugging that Merck is currently hiring. If anyone would like to contact Anya and I to learn more about opportunities here at Merck, please do. And I guess I'll thank all of our speakers one more time for the great talks and participation in the panel. So thank you so much, everyone. Hope you have a great rest of your day. Thanks.
Thank you so much, everyone. It was a pleasure to talk to you all, and thank you so much for sharing your science and technology with us.
Our pleasure. Bye-bye.
Thank you.
Hey, Anya, I think you're gonna have to stop the live stream, or you can promote me and I'll stop it.
Okay, hold on. How do I do it? Make a host.